Okay, first of all, I'd like to thank GAP for hosting this luncheon and providing the wonderful food and the place that we can meet. I'd also like to thank Amanda for all of her awesome work and advocating for the Food Ingredient Health Research Institute and for being my attorney and representing me as a whistleblower. She's been right there for me for 5 years now, maybe even longer. And I've come all the way from Hawaii, which is a really long way away, and today's lecture is a honor and privilege to be here. You'll be the first to see what I've been working on for a couple of years, which is a macroepigenetic model that explains insulin resistance. There's so much information out there now, widely available, so many journal articles. I mean, the truth is out there. There's just no arguing about it at this point if you really read the references that are available. So anyway, I'm looking forward to publishing this paper. And the last time I came last year, I had a different model for autism, and that one has been published. And this year, let's see, it was in April in the Clinical Epigenetics Journal, and we've had 116,000 downloads. It's the most viewed article published by Biomed Central, and they publish hundreds of medical journal articles. So it's a pretty successful model, I think, if people are reading it at that extent. So with that, I thank everyone for your support on food ingredient safety. We couldn't do the work without you. Okay, so I want to tell you a little bit about our mission at the Food Ingredient Health Research Institute. We are a 501c3 nonprofit, and our mission is to promote and support research and education on food ingredient safety for the purpose of disease prevention. We currently host an online nutrition intervention course for individuals seeking knowledge on nutrition and food ingredient safety through the study of gene-environment interactions. We teach using a macroepigenetic approach. Students wishing to earn 3 units of college credit in nutrition science may enroll in the course at Fort Peck Community College in Montana. It's online. Our organization is currently seeking funding to expand our research and education efforts so that we may reach more citizens. We do not accept donations from supplement, pharmaceutical, chemical, or any other industries associated with food ingredients, chemicals, supplements, drugs, or snake oil of any kind. We all know who I am, and I'm a retired United States Public Health Service officer. My expertise is in toxicology and environmental health. I told you a little bit about our most recent publication. We've published a number of different articles. The current article is a macroepigenetic analysis of the factors most responsible for the autism epidemic. So that was the article that folks are reading right now. So what is macroepigenetics? Macroepigenetics is defined as the process of examining food supplies and their impact on body metabolism and gene function, along with what is known about environmental exposures across populations. Today we will be the article that I mentioned with the 116,000 downloads. In that article, we used the macroepigenetic approach to compare variations in diet and toxic substance exposure between populations in Italy and the United States to determine the likely factors responsible for the autism epidemic in the United States. Today, we will be taking a macroepigenetic approach as we explore the factors responsible for the diabetes epidemic in the United States. So CDC says you're at risk of type 2 diabetes if you are 45 years of age or older. And I'm not going to read all of these things, but why don't you take a few minutes to read them? And then I'm going to ask you how many risk factors. Does anyone here have more than 2 or about two risk factors? So, does anyone have 2? I have. Okay. So these are a lot of risk factors. A lot to worry about. How many people here think that this is all, that this is it? This is it for risk factors. There's more, but they're not on this list. Let's see what's missing from the CDC list. Here's what's missing. The risk factors not listed are what we eat, how much food we eat, nutritional imbalances caused by the food we eat, and how what we eat determines how our bodies will manage exposures to environmental toxins that create conditions for the development of type 2 diabetes. That's what's missing from the list. Nobody talks about that. Obesity is an important risk factor for type 2 diabetes. I think we can all agree that obesity is clearly related to food intake in most situations. Most people think that the more we eat, the more obese we will get. It turns out, however, it isn't how much we eat, but what we eat that makes us obese. I myself lost 30 pounds recently and can now eat as much as I want and not gain any weight as long as I refrain from sugar consumption of any kind. No sugar. Why do we need to eat? A lot of people don't really know why we need to eat. Aside from feeling hungry, you're hungry, you eat. Well, we need to eat so that our cells can have glucose so they can run properly. In response to food, the glucose level rises in our blood. As blood glucose levels reach a certain point after a feeding, insulin is released by the pancreas to let the cells know that glucose is available in our bloodstream. Think of each cell as having a door that opens to let glucose in when insulin raises a red flag. If we are in the habit of eating too much sugar, we can develop insulin resistance. Insulin is signaling with a red flag, but the door to the cell stays shut and won't let the glucose in. This is when insulin is no longer effective at helping our cells gain access to the glucose. And then our glucose levels build up in the bloodstream. Depending on what we eat, food intake may be controlled by insulin if our bodies are not resistant to it. If we eat too much sugar over time, we may develop insulin resistance, which leads to the development of type 2 diabetes. Once we are resistant to insulin, we can become obese. Sugar consumption is a key factor in the development of obesity, according to Dr. Rob Lustig, a world-renowned pediatric endocrinologist at the University of California. And at the end of this presentation, there are several slides that there's a link to his presentation, a 90-minute video, and it is awesome, so I encourage you to watch it. But which kind of sugar makes us fat? High fructose corn syrup or cane or beet sugar? Well, excessive consumption of both kinds of sugar can make us fat, but not at the same rate. The rate of obesity depends on the amount of fructose we get from our sugar. The more fructose we consume, the quicker we gain weight. It all comes down to the fructose-glucose ratio in the sugar molecule. Every cell in our body can metabolize glucose, but only the liver can metabolize fructose. Sugar from sugar cane and sugar beet has a fructose-glucose ratio of 1.0. This means for every unit of fructose consumed from cane sugar, we consume 1 unit of glucose. Sugar from high fructose corn syrup, on the other hand, is more fructose than glucose. High fructose corn syrup 90 has a fructose-glucose ratio of 18. This means 18 fructose units for every 1 glucose unit. High fructose corn syrup 55 is the most frequently used fructose in soft drinks, and it has a fructose-glucose ratio of 1.34. This means we are getting more fructose than glucose every time we consume a soft drink. When we consume high fructose corn syrup in a soft drink, excessive fructose enters our liver, and it is converted to fat. Over time, if we keep consuming high fructose corn syrup, we will eventually become obese. We will become obese if we eat too much cane sugar also, but not as quickly. The whole process of becoming fat is complicated from a biochemical perspective. What does high fructose corn syrup consumption do to our bodies? High fructose corn syrup consumption leads to phosphorus and zinc loss in humans. Phosphorus and zinc are required by the body to regulate genes that control food intake. Insulin itself is able to regulate the transcription, the turning on, of specific genes. Without adequate amounts of phosphorus and zinc, our body does not know when we've had enough to eat. Too much fructose consumption leads to obesity quicker and the development of conditions such as non-alcoholic fatty liver disease, metabolic syndrome, insulin resistance, and type 2 diabetes. Metabolic syndrome refers to a set of risk factors that occur together and increase the risk of coronary artery disease, stroke, and type 2 diabetes. These risk factors are central obesity, insulin resistance, elevated blood glucose, high blood pressure, elevated triglycerides, low HDL cholesterol, which is the good cholesterol. In laboratory studies, researchers feed rats fructose to promote the condition of metabolic syndrome. Just how much of this toxic substance do we consume in the U.S.? I want to ask a question. Who knows what a toxic substance is? Raise your hand.
A toxic what?
Toxic substance.
Poison.
Poison? Something that makes you sick or kills you? Yeah. So does it matter if you get sick today or if you get sick 10 years from now?
I vote for 10 years.
10 years from now? So a lot of people argue about, well, it can't be that toxic if, you don't die for 10 or 15 years, right? But, you want to be hooked up to a kidney dialysis machine or something? Is that a pleasant way to go? Or need a kidney transplant 10 years down the road? If you're going to die eventually because of consuming something, then, too much of something, let's say, then some people might argue that point. I'm sure the corn refiners think that high fructose corn syrup is wonderful. Okay, so let's see how much high fructose corn syrup Americans eat. Well, from the table here we see that Americans have increased their overall sugar consumption by 21% since 1975. Right here. Okay? In terms of what kind of sugar they are consuming, they have increased their high fructose corn syrup consumption by 920%. That's a big shift on the type of sugar. Okay? And decreased their cane and beet sugar consumption by 29%. Wow, that's a big shift. High fructose corn syrup is by definition a high fructose sugar substitute. By consuming so much fructose, we are making our livers work overtime. What does our liver do? What does it do? It
breaks down the toxins in our body.
It breaks down the toxins. Yeah. So when all this where is all this fructose most frequently found? How can we become exposed to this substance? High fructose corn syrup is found in hundreds of food products, but our greatest exposure comes from the consumption of soft drinks or sugar-sweetened beverages, SSBs. Is there a link between the consumption of soft drinks and obesity in the U.S.? Yes. The weight of the epidemiologic and experimental evidence indicates that a greater consumption of sugar-sweetened beverages is associated with both weight gain and obesity. There's a lot of studies that have already shown this. There's nobody that's going to argue that fact at this point. Is there a link between the consumption of fructose and the development of insulin resistance and other elements of the metabolic syndrome? Yes, there is a link between the consumption of fructose and other sugar-sweetened beverages and the development of insulin resistance and type 2 diabetes. Every year, more and more studies come out. We don't see studies saying, no link. You know, you don't hear about studies saying, no link's been found. They're all saying there's a link. What does high fructose corn syrup consumption do to people in other countries? Well, according to a recent study published in the Global Public Health Journal by Dr. Goran and his research team in 2012, type 2 diabetes prevalence is 20% higher in countries where there is higher availability of high fructose corn syrup compared to countries with less access to high fructose corn syrup. This higher type 2 diabetes prevalence was found to be independent of the obesity risk factor. What this means is people with greater access to high fructose corn syrup are more likely to develop type 2 diabetes whether or not they are obese. With all of this background information under our belt, we are now ready to do our macroepigenetic analysis of the factors likely responsible for the type 2 diabetes epidemic in the U.S. We'll accept the idea that obesity is a major risk factor for the development of diabetes and use the macroepigenetic approach to analyze the difference in the diabetes prevalence between Germany and the United States. From our chart here, we see that Americans are clearly more obese than Germans and have significantly more diabetes. 35.5% of American men were obese between 2009 and 2010, whereas in the same time frame, only 23.3% of German men were obese. 35.8% of American women were obese in America, or the United States, obviously, whereas 23.9% of German women in the same time frame. 11.8% of American men have diabetes compared to 7% of German men. 10.8% of American women have diabetes compared to 7.4% of German women. As a population, Germans are less obese and have less diabetes. Now here we have a nice slide prepared by the Center for Disease Control. And it shows in the orange here the number of people with diabetes in the United States in millions. And then in the yellow, it's showing the percentage of the population with diabetes in the United States. So what it looks like is in the mid to late 80s, then there was things started moving upward, an upward trend. This is where it really takes off. That's about when high fructose corn syrup saturated the market, right there. See that? It's going up. In our most recent research, which is not yet published, we looked at what I haven't tried to publish it yet because I haven't written the manuscript, but I will. And we will. It will publish. So anyway, but here's what we looked at, what Americans eat on an annual basis and how what they eat might contribute to the development of insulin resistance and type 2 diabetes. We also looked at what Germans eat. We looked at what both of these populations are likely exposed to in the environment, possible gene variation, and then compared their type 2 diabetes prevalence. Americans eat a significant amount of high fructose corn syrup in their diet. Germans do not at all, because high fructose corn syrup is not readily available in Germany due to trade restrictions. Like only 5% of their entire sugar can be high fructose corn syrup in Germany. They just don't have access like we do. So in 2010, each American ate 37.8 pounds per year of corn sweeteners, most of it which was high fructose corn syrup. The only major difference between what we eat and what the Germans eat is the fructose. So what is it about fructose that makes our bodies become insulin resistant and we develop type 2 diabetes? How does high fructose corn syrup consumption lead to insulin resistance and type 2 diabetes? There are so many mechanisms. Let me count the ways. But today we're only going to go over a few. When we publish our manuscript, it'll be quite detailed and technical and cover at least 6 mechanisms. Consumption of high fructose corn syrup by Americans leads to phosphorus losses when dietary magnesium intake is low. This is unfortunate since dietary intake of magnesium is lower than it should be in the American population according to CDC researchers. So the American population overall is probably experiencing a phosphorus deficit in their diet from a macro point of view. Phosphorus is a key micronutrient because, as I mentioned previously, it plays a role in turning the genes on and off that insulin regulates, which is an important factor in diabetes and obesity. The German population, on the other hand, does not have a dietary magnesium insufficiency, even in their diabetic population. So they are probably not experiencing a phosphorus deficit as a group. A bit more on the crucial role of phosphorus and why our bodies need to hang on to it. Phosphorus depletion is also associated with decreased levels of ATP, which means adenosine 5-triphosphate, which is a molecule. It's an important molecule as it transports chemical energy in cells, and it's dependent on phosphorus. You got to have phosphorus to make it. Intracellular ATP depletion causes insulin resistance. Okay? Look at the dates of these studies, 1978, 1980. They're pretty old, aren't they? Well, let's look at some new studies. Here we are. We have some very current studies, 2011, 2012, published in the last 2 years that show the connection between fructose consumption and low ATP activity in humans that are insulin resistant or obese and have type 2 diabetes. Liver ATP synthesis was found to be lower and related to insulin resistance in patients with type 2 diabetes in one study. Higher dietary fructose was associated with impaired ATP homeostasis in obese individuals with type 2 diabetes in another study. The connections seem quite clear to me. Do they seem clear to you? Everyone? Am I going too fast? No? Got it? All right. This is just one pathway that consumption of high fructose corn syrup leads to insulin resistance in type 2 diabetes in Americans. There's another pathway we will talk about today, and that is the role that mercury plays in the development of insulin resistance. It may come as no surprise that the intestinal absorption of phosphorus can be blocked by mercury. We've now got to talk about mercury exposure as a factor in the development of insulin resistance in type 2 diabetes. Is there any evidence to suggest that mercury exposure in humans leads to insulin resistance or type 2 diabetes? Yes. In 1,449 non-diabetic residents living near a mercury cell chlor-alkali factory in Taiwan, researchers found that those with higher blood mercury levels were at significantly higher risk for insulin resistance. In another study of 4,582 Americans who participated in the National Health and Nutrition Examination Survey, those with low-level mercury exposures had a greater risk of unexplained liver disease. Most of the subjects were insulin resistant. In both of these studies, blood mercury levels were used to determine mercury exposure. It didn't matter if mercury levels were low or high. Mercury exposure was associated with the development of insulin resistance. There is an interesting difference between German and American populations when it comes to mercury exposure and blood mercury levels. Here we see a spatial distribution of global anthropogenic or man-made emissions of mercury to the atmosphere. Here we see the lighter blue in the United States, and over there in Germany and Europe it's dark blue. That means there's more mercury in their air, the air they breathe. So in Germany the population is exposed to greater atmospheric mercury levels than Americans, yet their blood mercury levels are lower. Despite greater exposure to environmental mercury, German blood mercury levels are lower than those found in the American population. Even with fish consumption, the geometric mean for blood mercury levels in the German population is lower at 0.77 micrograms per liter than the mean for blood mercury found in the United States, which ranges from 0.94 to 1.06 micrograms per liter. Germans do not consume high fructose corn syrup. Could this difference in blood mercury levels between geographical populations indicate that consumption of high fructose corn syrup may play a role in mercury metabolism, or perhaps contribute to overall mercury exposure. Yes, high fructose corn syrup interferes with heavy metal excretion by disrupting metallothionein function. Consumption of high fructose corn syrup leads to zinc loss and copper gain in humans. Zinc is needed by the body to build the metallothionein transporter protein. This protein carries heavy metals out of the body. Excessive fructose consumption, therefore, may interfere with metallothionein function and compromise the body's ability to excrete mercury. This interference explains why Americans have higher blood mercury levels than Germans. Consumption of high fructose corn syrup by the American population creates a mechanism for mercury bioaccumulation. Do you see that? Does it make sense coming together? Consumption of high fructose corn syrup can also be a source of mercury exposure. How can high fructose corn syrup be a source of mercury exposure? Corn refiners have historically used mercury cell chlor-alkali chemicals to make their product, and as a result, mercury has been found in high fructose corn syrup and products containing high fructose corn syrup. 3 different studies, by different research teams have found mercury contamination in high fructose corn syrup and products containing high fructose corn syrup. The most recent findings were reported by Canadian scientists led by Karen Rideout in 2010. At the beginning of this presentation, we mentioned that consumption of high fructose corn syrup can lead to phosphorus loss in humans. We know this because USDA researchers did a study on humans who consumed high fructose corn syrup. Why does consumption of high fructose corn syrup lead to phosphorus loss? Well, it could be related to the mercury content in high fructose corn syrup. Mercury blocks the intestinal absorption of phosphate. When phosphate cannot be absorbed by the body, then it's going to be excreted in the urine or the feces. This excretion of phosphorus can, as we mentioned before, lead to problems with ATP synthesis and insulin gene regulation. Am I going too fast? No. Is there additional evidence to suggest that mercury exposure can lead to the development of insulin resistance in the U.S.? Yes. Researchers continue to find that low-level mercury exposure can affect thyroid hormone status, and that this is yet another mechanism for the development of insulin resistance. This is bad news for Americans because blood mercury levels in the American population appear to be rising. A UCLA researcher by the name of Dan Lax looked at the data collected by CDC's National Health and Nutrition Examination Survey and found the percentage of American women with detectable inorganic mercury in their blood has risen astronomically. Within the population of women of childbearing age, inorganic mercury detection rose sharply from 2% of women in 1999-2000 to 30% of women in 2005-2006. Coal-fired power plants are the largest sources of unregulated mercury exposures in the U.S. Mercury emissions have not historically been regulated in the U.S. or Germany. Now we're going to have regulations soon. Someday they're going to be in effect. What about mercury in fish? The studies we talked about that showed associations between blood mercury levels and insulin resistance assumed that fish consumption was the major source of mercury exposure. What do we know about the toxicity of mercury from fish consumption? The toxicity of mercury from fish consumption depends on dietary selenium intake. You need enough selenium in your diet to support glutathione production. Glutathione is another protein needed by the body to excrete mercury. The glutathione oxidation reduction system needs phosphorus to work right. Does anyone remember what food ingredient leads to phosphorus loss? High fructose corn syrup. Does anyone know what foods are high in selenium? Brazil nuts. Is the solution to the mercury in fish problem to avoid eating fish or to eat more Brazil nuts? Here we
What
have.
is it?
Well, let's keep reading here. Here's a nice chart provided to me by Dr. Caitlin Hafey, the author of the Mercury Study Report to Congress that was many years old now. Anyway, but the dark black lines going up is mercury. And so the fish with the least amount of mercury is salmon, right over here. And the fish with the most amount of mercury looks like snapper, red snapper, maybe sea bass, and tuna. We don't need to avoid fish. Fish is a wonderful food that supplies the body with essential omega-3 fatty acids required for maintaining neuronal plasticity and learning capacity. In a recent study, researchers collected hair samples for mercury analysis and fish consumption data from mothers during pregnancy. The researchers followed up with the mothers several years later to find how their children were doing. They determined that the fish consumption during pregnancy can protect against attention deficit hyperactivity disorder as long as mercury exposure is low. Fish has many nutritional benefits. The omega-3 fatty acids available in fish also play a role in the prevention of obesity. So, the solution to the problem of mercury in fish is to eat fish low in mercury. Here we see mercury levels in different species of fish and shellfish. Salmon is the best fish to eat in terms of reducing your mercury exposure. Mercury levels are relatively low in crab, clams, trout, shrimp, pollock, and catfish, as can be seen in this chart. So again, fish is an important food to eat as it provides essential omega-3 fatty acids required for maintaining your brain cells and learning capacity. We call good brain cell health neuronal plasticity, which is how well you can keep your memories, okay? We won't even talk about Alzheimer's. I have a macroepigenetic model for Alzheimer's too, but that'll be another year down the road. I said that twice, didn't I? Omega-3 fatty acids must be important. Yeah, said that twice. Omega-3 fatty acids are unsaturated fats found in fish and plant oils like flaxseed. So here we have a little visual for you. And there is strong evidence that the omega-3s, EPA and DHA, which are very long names, chemical names, can boost heart health and lower triglycerides. As you may recall from an earlier slide, elevated triglycerides is a risk factor for diabetes. We do not want to have elevated triglycerides, so we need our omega-3 fatty acids. Zinc is required by the body to produce omega-3 fatty acids, EPA and DHA. Consumption of a zinc-depleting substance such as high fructose corn syrup leads to a reduction in omega-3 fatty acids. Does that make sense? Everyone with me? And with overconsumption of omega-6 fatty acids, which are like vegetable oils, salad oil, then what happens is an increase in the omega-6, omega-3 fatty acid ratio. Omega-6 fatty acids are found in margarine, vegetable oils, cracker, cookies, and other processed foods that contain hydrogenated soybean, sunflower, and corn oils. Hydrogenated oils are oils that are chemically extracted from vegetable seeds. Contrary to what most Americans have been taught, fish is not the only way humans obtain the important omega-3 fatty acids, DHA and EPA. The human body can actually produce its own omega-3 fatty acids, DHA and EPA, from the essential fatty acid known as ALA. The conversion from ALA to EPA and then to DHA is often insufficient in humans because of the Western diet, high in omega-6 fatty acids. Humans evolved on a diet with a ratio of omega-6 to omega-3 of 1. So for every one omega-6, there was one omega-3. That's how we evolved. With the adoption of a Western diet, the ratio has increased to 15 omega-6 to 1 omega-3. In order to successfully convert the essential fatty acid ALA to EPA and then to DHA, one would have to stay away from processed foods that lead to mineral losses like zinc losses, and instead eat foods that provide enough of the correct nutrients required to support the conversion process. These important nutrients include magnesium, zinc, vitamin C, B6, and B3. I know this is way more information than you probably want to hear. Is your head spinning? Are we doing the exorcist? I know it's a lot. Okay, here's the macroepigenetic model for insulin resistance and diabetes. And we have in the middle mercury and or high fructose corn syrup. Doesn't matter, either one. And then, okay, so I'm just going to go through it and try to follow the arrows. But you see all the mechanisms here from high fructose corn syrup leading to insulin resistance? There's so many mechanisms. It's amazing. Okay, so here we go. Humans are exposed to mercury, have detectable mercury in their bloodstream. Okay, so a recent study showed that humans living near a chlor-alkali plant had elevated blood mercury levels, and those humans developed insulin resistance. Okay, you see that? Well, actually, they had a increased risk, significantly increased risk of developing insulin resistance. Then we noted earlier that mercury cell chlor-alkali or chlorine chemicals have historically been used to manufacture high fructose corn syrup. Consequently, mercury has been consistently found in high fructose corn syrup and products containing high fructose corn syrup. So dietary mercury prevents phosphorus absorption by the intestinal system. So then that leads to phosphorus loss and zinc loss too. Phosphorus is required by the body for the processes that regulate genes that control food intake, insulin function, and excreting mercury. So then you get a loss of phosphorus required to regulate genes that control food intake, and then you get obesity, or you get insulin resistance, and then you get type 2 diabetes. Oh, and then there are studies showing that parents that have type 2 diabetes then give birth to children with insulin resistance. So we have a transgenerational effect, you know. So that means the more and more we eat high fructose corn syrup, the more insulin resistance we're going to have in the next generation. That's what we're doing to our children. Let's see. Okay, so... Oh, a recent 2010 human study showed that dietary phosphorus is required to control appetite and food intake. Phosphorus loss leads both to insulin resistance and obesity. Insulin resistance is associated with the development of type 2 diabetes. With everything we know so far about gene regulation, it is clear that environmental mercury exposure along with consumption of high fructose corn syrup leads to obesity and insulin resistance, and later the development of type 2 diabetes. With a true epigenetic model, what we often see is dietary changes in one generation affecting the health of children in the next generation. In this model, that is exactly what we see. In the case, like I said, with parents with type 2 diabetes who give birth to insulin-resistant children. There's another route to insulin resistance involving gut bacteria, but I'm not going to cover that today. But just know that there are several mechanisms associated with fructose. Okay, so today we've learned why we are what we eat. It makes sense that if we have an obesity and diabetes epidemic in the U.S., then Americans are eating an awful lot of foods leading to phosphorus and or zinc loss and omega-6 gain. The average American is eating 37.8 pounds per year of corn sweetener, which is primarily high fructose corn syrup. Consumption of high fructose corn syrup leads to essential mineral imbalances, including phosphorus and zinc loss. Zinc loss is detrimental in maintaining the omega-6, omega-3 fatty acid balance. Phosphorus loss is detrimental in regulating insulin and food intake. From our chart here, we see the average American consumes 36 pounds per year of salad and cooking oil. Those are the omega-6. The most commonly used cooking oils contain a significant amount of omega-6 and very little omega-3. By consuming so much omega-6 fat and zinc and phosphorus depleting high fructose corn syrup, the average American is increasing their risk of becoming obese. Germans, on the other hand, are less obese and have lower type 2 diabetes primarily because what do they not consume? They don't consume high fructose corn syrup. Today we have used the macroepigenetic approach to explore the factors responsible for the type 2 diabetes epidemic in the United States. We looked at variations in diet and mercury exposure between populations in Germany and the United States to determine the likely factors responsible for the development of insulin resistance in the United States. We learned that consumption of high fructose corn syrup leads to mineral imbalances that impair our body's ability to regulate glucose and insulin and produce important proteins essential for heavy metal excretion, thus making us more susceptible to developing insulin resistance and type 2 diabetes. We learned that we have a big problem that needs to be addressed. What is the Food Ingredient and Health Research Institute trying to do about the problem? We have developed a protocol to use in conducting a study to determine if consumption of high fructose corn syrup leads to the bioaccumulation of mercury and the development of insulin resistance in humans. We already know that it leads to insulin resistance, but we need to be able to show that along with the insulin resistance comes the bioaccumulation of mercury. So the way we want to do that, well, we've recruited collaborators in communities in 4 different regions of the United States. These collaborators are 5 different tribal governments, a Native Hawaiian community, healthcare providers, and researchers at 2 community colleges and 2 major universities. We have collaborative agreements in place for biomarker testing for the participants involved in the proposed study. In our study design, half of the participants will refrain from high fructose corn syrup consumption, and the other half will continue to eat the standard American diet over a 3-month period. We will collect pre- and post-intervention biomarker measurements to include, at a minimum, blood mercury, insulin, and glucose. The participants who refrain from the consumption of high fructose corn syrup will be asked to participate in a weekly support group and will receive a small monthly stipend to help them buy alternative foods during the 12-week intervention period. They may also participate in the online macroepigenetics course and receive 3 nutrition science credits from the Fort Peck Community College. The participants who continue to eat the standard American diet will be eligible for the monthly stipend and may enroll in the online macroepigenetics course after the initial 12-week intervention period. The pre- and post biomarker measurements will allow us to determine the role high fructose corn syrup plays both in the bioaccumulation of mercury and the development of insulin resistance. The study itself will take 4 years to complete across 4 geographic regions. With 60 participants in each region, we will have 240 participants. We need that many participants so we'll have the statistical power to show the significance of our findings. When will the study be done? The study will not even get started until we get some funding. Since high fructose corn syrup is considered safe by the FDA, it is unlikely we will get funding from the FDA or any other Department of Health agency. How is the government going to look if we find and show that high fructose corn syrup plays a definitive role both in the bioaccumulation of mercury and the development of insulin resistance? This is not going to be popular. Such a finding would demonstrate definitively that high fructose corn syrup is a toxic substance. The goal of the study design is not likely to be supported by any conventional organization in the United States. We have developed a responsible budget for this proposal, and we know it will cost $2.5 million. We have determined the costs associated with paying the participant stipends, tuition, biomarker testing, travel to and from the 4 study sites across the country, nutrition intervention training for the community co-investigators, institutional review board review and approval, and so on. It is expensive to conduct medical research. Although the cost seems like a lot, I think I might have, yeah, okay. Although the cost seems like a lot, our study design is excellent. There is no doubt we will be able to determine the role high fructose corn syrup consumption plays in the bioaccumulation of mercury and the development of insulin resistance. We have our collaborative agreements in place. With your help, we could get the ball rolling. That's the public's help, because we're not going to get the funding from the powers that be. This study is not going to benefit anyone in our economic system except for the consumer. It's not going to benefit anyone else. Okay? So that means the consumers have to pay for this. No one else is going to help. So it's whether or not the consumers want to know this. Is the high fructose corn syrup making us bioaccumulate mercury, and is this a factor in the development of insulin resistance and diabetes? And does this mean that we are passing on these diseases to our children because of the foods we're eating today? You know, this is serious business. What can you do to help? You can give a tax-deductible donation to the Food Ingredient and Health Research Institute at the link provided. We cannot finance the study without your support. Please give what you can. If everyone donates a little, we will be able to raise the money we need to achieve our research goal and complete the study within a 4-year time frame. Please share this presentation with your friends and family and post this link on your Facebook. We can't do it without your help, and no one else is going to fund us. So here are the take-home facts, just a few facts to take home with you. The evidence suggests that high fructose corn syrup is a toxic substance because it impairs our body's ability to regulate glucose and process and excrete mercury and other heavy metals. Our children, yours and mine, are increasingly at risk of becoming obese and developing insulin resistance and type 2 diabetes. As we are being exposed to more mercury, it is increasingly being stored in our bodies, just like other mammals. Okay. So this is just a picture of the model we already published about the factors responsible for the autism epidemic in the United States, and it's not a hard paper to read, so if you want to investigate the causes of autism, you can check it out. It's at that link. And it's pretty consumer friendly. Are mercury, high fructose corn syrup, and organophosphate exposures associated with the development of autism or the prevalence of autism in North America? Yes, yes, yes. And that's why 116,000 downloads so far, you know. Here are the latest figures for the U.S. Department of Education, the numbers of children receiving special education services. And so the number of children since 2005 to 2011, the percent change is 110% increase, autism. For other health impairments such as ADHD, attention deficit hyperactivity disorder, from 2005 we've had a 31% increase in the number of children served. And then developmental delay, which is another condition that often looks a little bit like autism, 46% increase. And then even though all of this increase, there's been a 5% decline in overall special education cases served. So that means fewer children overall with special education services being served, but way more of the children who are being served, continual increases in autism, ADHD, and developmental delay. And that's a direct relationship with the food we're feeding our children, you know. And it's all about the food. Does anyone have any questions?
Thank you for that very informative talk. I have a question. I have a few, but I'll just ask one and then open the room. What is the difference between high fructose corn syrup and corn syrup. And Amanda did some research through you on this and sent it to me, but I think it could be informative for the room. Because I see labels of things that say no high fructose corn syrup, and then it still contains corn syrup.
Okay. My understanding is that it's in the same product line, you know, so you have your corn, and then from that corn, there's a whole process that they have. They make all these different products, you know, like cornstarch, dextrose, you know, different corn sugars, and then at the very end of the product line is high fructose corn syrup. But corn syrup's in there somewhere.
Mm-hmm.
It just isn't, you know, it's not the total end product.
Mm-hmm.
That's my understanding.
So the trace levels of mercury there isn't research to demonstrate it's in corn syrup per se.
I haven't seen a study on that. We haven't, there's been 3 studies, and the Canadian group looked at products on their shelves in 2010 of ingredients that contained high fructose corn syrup, first and second ingredient. And they didn't just, you know, it wasn't differentiated out to like corn syrup.
Mm-hmm.
So, you know, I wouldn't presume that the corn syrup did not contain mercury. Mm-hmm. You know, just like there are allowable levels of mercury in specific food colors, mm-hmm. like yellow 5, 6, mm-hmm. allowable levels of mercury in annatto, which is a food that color in butter, and allowable levels in carrageenan, which you see in ice cream. There are allowable levels of mercury in a lot of food ingredients that are used for preservation purposes or coloring. So we, you know, there's allowable levels of mercury in flour that's been chlorinated, white bleached flour. So I would not presume that corn syrup doesn't contain mercury, given that it's, you know, probably who knows? You know, we can't really say for sure, but there's some intent, I think. There's intent to use whatever chemicals will maximize product shelf life. Because the longer a product can stay on the shelf, then the more you can transport it to, you know, it can go to China, and it can stay on the shelf for months and months and months, and sometimes longer. Twinkies, I don't know. You know, there's some products that I don't even know if they have a shelf life. So, yeah, I don't even know. I think Twinkies are made up of nothing but chemicals, you know. So, all the chemicals in the world come from the foundational chemicals. So the chloralkali chemicals, chlorine, sodium hydroxide, hydrogen chloride, and potassium hydroxide, from those chemicals come all the other thousands and thousands and thousands of chemicals. So when they make the 4 baseline chemicals with mercury, which they do still, maybe not so much that they make it here in the U.S., because I think there's only 4 mercury cell plants left. It doesn't matter because we import it. We import those chemicals with the mercury grade, mercury residue. We import it from the United Kingdom because they make a whole bunch of it there. And so the U.S. is really, you know, we just, even if we were to outlaw mercury cell chloralkali plants here, we would, the food industry and the pharmaceutical industries would still get those chemicals from the United Kingdom. They would just import them. So it is what it is. We have a system of food manufacturing that relies on chemicals for food preservation, product shelf life, because these chemicals prevent mold in your bread. That's why you have fructose in your bread. I mean, it's product shelf life. Why would you put some of these chemicals in the food to begin with? It's so that you don't have mold, mildew, bacterial growth, and all of that. So, you know, we can buy local, have local foods, or local produce, farmers markets, and stay away from processed foods. That's your best bet. Did I answer your question?
Early in your presentation, you were illustrating the ratio between high fructose corn syrup and glucose. And there was one type of high fructose corn syrup that had like a 134 to 1 ratio. And there was another that was 1.8 to 1.
Or
So I
18.
think the first one's
Yeah.
a lot worse.
Yeah,
But how do we tell the difference?
The high fructose corn syrup dash 90
Uh-huh.
is not used in soda, soft drinks.
Uh-huh.
Yeah, that's used in different products. So that, I'm not sure which one that's used in. That might be the one that's used in the, like the bread and stuff, you know.
Uh-huh.
But the high fructose corn syrup 55, that's the one that's in the sodas that we consume so much of.
Is that the one that's 1.8?
1.34.
1.34.
Yeah, the other one wasn't 1.8, it was 18.
Oh, I see.
18 fructose units for every 1 glucose.
But you're not sure what product
I don't
had
know,
that one?
yeah. But it would be worth finding out. That's an excellent question, because with that much fructose units, maybe you don't need as much of it in order to have a debilitating effect. You know, that's a really good point.
Another, some points you were making, that the high fructose corn syrup undermines our levels of needed minerals like selenium or zinc or phosphorus. Can we compensate for that by going to General Nutrition Center across the street and taking selenium and zinc and phosphorus tablets to compensate for processed food?
That's a tough question, and the reason why is because the FDA only started regulating those supplements.
Uh-huh.
And I don't know how far along they are in their regulatory process.
Uh-huh.
But some supplement companies have testing on them to see that there are no trace contaminants of heavy
Uh-huh.
metals like mercury or cadmium or whatever. But they don't all do that. So we don't really know, you know, just because something says that that's vitamin B12 or selenium, there's, you know, how do we know that there isn't some contamination in there? Last year when I did this talk here, there was a physician in the audience who was taking a lot of supplements, and his he got a toxic metal overload, and his physician asked him where he, they figured out he was getting it from the supplements. So if you're going to take supplements, then you would want to take supplements that have been undergone some standardized testing to make sure that they don't consume any chemicals or elements, heavy metals, cadmium, whatever, that you wouldn't want to be ingesting in your body.
Now, would organic versions of these products be presumptively safe, or they need testing too?
I would look for something that has undergone some testing or certification. That if I were going to look for a supplement. But really, the best thing you can do for your children, small young children, developing minds, is to feed them whole foods, fresh fruits and vegetables, preferably organic, but if not, very well rinsed to reduce their pesticide exposures. But it's the whole foods, like the spinach. Back in the day when we had Popeye and how he ate his spinach. I mean, there's no lie. I mean, that chart I showed, I should have gone over it in more detail. We eat less than a pound a year. I think it was less than a pound a year of spinach. That's a superfood. Magnesium and calcium, has all these wonderful things that our body needs. And then, close to, 38 pounds a year of fructose and 28 pounds, I can't remember how much vegetable oil, and then less than a pound a year of spinach. I mean, it's crazy. So they need their fruits and their vegetables. And you can make smoothies. You can make smoothies with almond milk and spinach and strawberries, and put a teaspoon of cod liver oil in there for their omega-3s and some 2 Brazil nuts or something. And wow, that'd be a wonderful little treat for a kid. And they're getting their spinach. They don't even know it, but they're getting it. And they need to have those dark green leafy vegetables, and they've got to have some, good fruits and fiber, and to lay off the sugars is much as
So
you
to
want.
follow up on that point, just because it's the same question, topic, how does collard greens or kale or other dark green vegetables compare to spinach? You only listed spinach.
Yeah, no, those are good too, the dark
Okay.
greens, yeah. Any dark greens are very good. Spinach is the superfood, I mean, you know, kale, all that stuff is
Okay.
good.
Obviously, it looks like, you know, we're hit our 1:30 mark 15 minutes ago. But do we have a couple more questions, Renee, if you don't mind?
Yeah, sure, I got plenty of time.
I was just wondering about the, if you addressed this earlier, I apologize for not hearing it, I came in late. But what do you think is the most effective action, assuming that the study does show that, you know, high fructose corn syrup is toxic? Is it consumer awareness or FDA regulation? You know, where would you go with that?
I wouldn't know where to, I'm more of a scientist. You know, we're turning towards advocacy out of, we have to go to the people, the powers with the people. The people have to want to do this, because the system that's in place is not going to do it, because there's no incentive in the profit-making system to change things. So what do we do if we find out what we think we're going to find out? And that would be, it'll take years of advocacy. I mean, when they found that lead in paint and lead in gasoline was bad for children, it took 20 years. I mean, the studies were done, like all the studies now that show fructose is toxic. And yet, you know, it'll be years before, and it will, the consumers have to be educated, and they have to demand that things change. And it's going to be education, education, education. We have to educate the consumers, and they have to want it. They have to want to have a different food supply. And they have to, enough people, I hate to say it, but I mean, it's about suffering. And then when 1 in 3 children are autistic, when, you know, at least one of your parents is diabetic and hooked up to kidney dialysis, I mean, how sick do we have to become before people want to address the issue? I mean, the food supply has been, for years, with the pesticides and everything else, there's been studies and more studies. And you know, people know what's going on. The consumers have to want to change things, and they have to become more active, because we can't do any more than we're doing. You know, you publish the studies, and you know, what more can you do? I don't know. Do you have a suggestion? What would you think? How can we make things safer for our children? My concern is as an adult, you know, what kind of world are we leaving our children? What are we setting them up for? What kind of health are we giving them? You know, so what do you think?
Well, I think consumer education has done a lot, as you suggested, with high fructose corn syrup. There's a lot of awareness.
Yeah.
So that might be the way to go, you know, because FDA regulation could take a really long time, or a ban. It'd be really tough, because the food lobby, food companies are really powerful. So
Right.
I think this is a great way to go about it.
You had peaches and wheat on your list in one of the later slides, and I wasn't sure if they were good or bad.
Well, I put them there because they could be a source of organophosphate exposure
Mm-hmm.
and pesticide residue, unless it's organic, which is a lesser risk of pesticide residue. So there's a role. I didn't go over all the roles, because there's so many mechanisms here in this. And so with the depletion of the zinc in our system, and the calcium, that we also have calcium depletion under certain conditions, then our gene that provides us with paraoxidase, which is a enzyme to break down the pesticides, the organophosphates, can be suppressed. And so the high fructose corn syrup can actually suppress that gene, and then we are not as able to process the organophosphates. So that we know that diabetics or people with insulin resistance have reduced PON1 gene activity.
Mm-hmm.
And that's probably a result of the fructose as well. What that might do to add to the insulin resistance, I don't know, but there's a relationship between that, that particular gene and what's going on there because of fructose consumption.
So those are two foods that could actually be threatening?
If they were not organic and if they were not washed,
it's because of the
yeah,
pesticide.
the pesticide residue would actually make things worse. It would make the situation even worse than it already is.
Now, do they stand out from other fruits that get pesticides?
No,
or it's
I
a
just
lesser
put, the
amount?
wheat is obviously, if we eat 95 pounds a year of wheat,
Yeah,
you know,
yeah.
they've already done a study, or a couple of them, that show that there is pesticide residue after you create an end product like bread. So, you know, baking it doesn't take it out, you know.
Okay.
So, yes, it will take out a lot, but it's not going to be completely eliminated.
So they were there basically to illustrate the threats from pesticides.
Yeah, there's a
Okay.
threat as well from that arena, but we didn't have time. I mean, to go over every aspect of how fructose impairs your body, you know, it would just be beyond the time that we have.
Anything, Charlie?
Well, I had one question, which is probably not helpful, but you had talked about diabetes levels in other countries, and I know like Thailand, for example, has a very high diabetes level because they put sugar in everything. That's probably why. But I was just wondering because I'm assuming they don't have high fructose corn syrup.
They probably, I don't know if they do or not. I looked up all the countries that we export fructose to, and I can't remember the list, but there's a bunch of them. So fructose is very common in a lot of countries now, and so they're developing the Western diseases, like type 2 diabetes.
They have historically had it there, I think, even though people are really skinny.
Yeah, well, you can still get it with sugar consumption. You don't, fructose just makes it happen quicker. But you can still get it with too much regular, you know, cane sugar. So.
I have a question from Twitter.
Okay.
What are your thoughts on like the food industry's ability to just, you know, replace high fructose corn syrup with like another cheap sweetener? Does it matter if they change to something that has similar amount of fructose or not, or?
Well, whatever they replace it with, then I think they should investigate the effects on humans before they do anything. I think what our system has been doing, which is probably not a good idea, is that they put things on the market before they've been thoroughly tested. And so then it's only after years that go by then, and plus with genetic variation, you know, I mean, 1 or 2 or 3 generations later, our bodies respond. We are like any other animal or plant. We respond to our environment, and our genes adapt. And it can be in a good way or a bad way. So I think that whatever sweetener they put in our food supply should be thoroughly tested before they put it in there. And I don't think we spend enough time. Before we rule something as safe, you know, we're ruling things as safe. There's a problem with that system that we have where we can rule things as safe. I mean, to get a substance generally recognized as safe by the FDA now, I believe the process is pretty simple.
Yeah.
And, you know, actually the manufacturer themselves can just basically submit information and say, hey, it's safe. And then FDA can say, okay, you know, we accept your data. So I think we need a third party agency who can be well funded by taxpayer money to do some third party studies on things. I don't think we should leave it to the manufacturers to provide their own data. I think that's, you know, when a profit motivated scenario, I mean, that doesn't serve the public health. I mean, that's insane. I can't imagine why we're doing this. Doesn't make any sense.
Anyone else? All right. Well, thank you so much, Renee, for joining us and being part of the social media experience as well. So, all right.
Thank you very much.
Thanks.
Thank you.